realise I haven't got my glasses, so sorry. Cool, getting old. Thank you. Well, at least with age comes wisdom. All right, so our next speaker, Dr Belinda Gray. So, with a passion for inspiring health and well-being, Dr Gray has been involved in the natural health industry for over 20 years. So she's an accredited naturopath, nutritionist, who remains committed to taking a holistic approach with diet and lifestyle and at the forefront. So through time spent in working in an integrative medical clinic, and with leading health practitioner-only supplement companies, Belinda became acutely aware of the need to improve evidence-based practice and education across the industry. So please welcome Belinda.
Slides? Yeah. I can talk to this, but my presentation would be far more helpful.
How do you talk to QR code?
I can talk about ANTA.
You could.
But it's not as much my area of expertise.
That one there?
Yeah.
Yep. Perfect.
Security alert. Yes, yeah. Oh.
So maybe enable.
Okay, so today's topic we're going to look at gut microbiome testing in clinical practice and the actual testing methods available. So before I start, can I get a show of hands for anyone who's actually either used microbiome testing in clinical practice or done some level of training in it? Fantastic. Those who've helped keep it up if you're actually using it still in clinical practice. Awesome. So I'm going to start very, very basic today. For those who haven't actually used microbiome testing before, for practice who trained probably over 10 years ago, it was never part of what we actually used. And what you will also see is that the availability of testing has actually changed a lot in the last couple of decades as well. So my background in this, obviously I've worked in research having done the PhD, but I also spent 2.5 years with Microba or Cobiome doing their scientific review and innovation prior to the launch of the Cobiome testing. So what is the gut microbiome? The microbiome is not just the bacteria in your gut. It does refer to the bacteria in your gut, but we also include things like the fungi, the viruses, your bile acids, the genetic material that's in your gut, the fibers and stuff that we're eating. Basically, anything that is in that large bowel area is what we are including when we're talking about the gut microbiome. It's not what we test for. We only test pretty much the bacteria or now some of the metabolites and stuff as well. But what we're referring to is that as a whole. When we look at how the microbiome is actually shaped, it starts off in utero, then influenced by the mother, influenced by the birthing process. That microbiome then matures as we age. It's influenced by our geography. So that includes everything from the fruit and vegetables and the foods we're eating to whether or not we're living in the suburbs or a rural area. It's influenced by our diet, pharmaceutical drugs, and no, I'm not just talking about antibiotics. Stress, exercise, lifestyle factors as a whole play a huge role in your microbiome, and we'll go into a little bit more detail on each of them later. Why is it important? Probably because it impacts our health. So the bacteria that actually exist in our gut produce different metabolites. And those metabolites, as you can see here, can have positive or detrimental effects on our health, and some of them can be positive to a degree unless we end up with too much of them, in which case they become detrimental. If, for example, you look at something like methane, it leads to slow intestinal motility and then constipation. If you go to the other side of the scale, some of the better bacteria will produce things like butyrate and IPA, associated with protection of the intestinal barrier and reduction of inflammation. So as you can see from that, it's not the bacteria, it's what they end up doing that actually has their impact overall on our health. What ends up happening is the long-term effect. From a gut-brain axis, we look at things like autism and neurodegenerative disorders. You can have associations between your gut health and atherosclerosis, heart failure. There's implications for the gut and the skin, so psoriasis, acne, skin cancer. There are then implications for the metabolism, so huge association with obesity and weight loss and type 2 diabetes, cancer, mitochondrial dysfunction. All of these can be tied back to the gut microbiome. If we take one of the specific examples would be hydrogen sulfide. We know hydrogen sulfide is associated with flatulence, pain, bloating, but what is it actually doing besides from that inside our body? It's produced by the bacteria in our gut. If we have excess of hydrogen sulfide, it can compromise our intestinal barrier. It basically splits the mucin disulfide bond and then inhibits butyrate uptake in the colon. Longer term, where does that actually lead to? ATP synthesis dysfunction, so we're not getting the energy production that we actually need inside the cells. We're also getting an increase in your inflammatory markers like IL-6, and I don't know why this is flicking over itself, and nitric oxide. So besides from just the bacteria and the metabolites, yes, they're important, but the other factor we really look into quite heavily when it comes to your microbiome is your microbial diversity. Now, when I use the term microbial diversity, I mean the type of bacteria, so the variability in different species within the gut, as well as the richness and evenness, so the abundance of each of those bacteria. And why is that important? If we have lots and lots and lots of different kinds of bacteria and we throw a challenge at them, so antibiotics or something like that, if there's lots of different kinds, the likelihood that good bacteria are going to survive enough for your gut to recover without significant symptoms is a lot better if you had a better, more diverse population to start off with. If you're starting off with just, say, 5 or 10 really good species, and all of those were susceptible to the same kind of antibiotic or the same kind of challenge that you faced, you then end up with a predominantly bad bacteria in your gut, and it's a lot more difficult for your gut to recover. So if we talk about increased microbial diversity, it leads to a more stable microbiome and better health correlations. If we have less diversity in our gut and we throw a challenge at it, we are more likely to end up with a reduced resistance to pathogens. There is an association with increased disease severity, and the one, Melbourne probably, it's a dirty word, COVID. There was a huge correlation between reduced microbial diversity and increased disease severity in COVID. There's a correlation between decreased microbial diversity and insulin resistance. There's also correlations with obesity, inflammation, and allergies. So one of the first things I look for when I get my patient reports back, if I'm using them for microbiome testing, is microbial diversity, because that's going to tell me, if my patient is thrown a challenge, exactly how well they are likely to recover from it. So testing methods. We've come a very, very, very long way since the 1600s. Even when I started clinical practice 20 years ago, the main options that were available to us were microscopy, giving it to a path lab to look at it under a microscope, and growing it on a plate. Now, whether or not we can cultivate it depends on whether or not it's still alive when it comes out. It depends on what we're actually trying to cultivate it on and the conditions we try and grow it in. 2 options are great, but nowadays we have many more options to work within. So the 2 main ones that you guys will come across are the 16S, which is like taking a tiny part of a DNA snippet and then saying, okay, well, exactly what bacteria are there. Or what they call shotgun sequencing, which is a slightly more in-depth version. To compare the different methods, the first two columns that you'll see there, the microscopy and culture, much, much cheaper. They will give us genus level, so the overall branch that the bacteria sit under. They will not necessarily give us the specifics of the species that we're talking about. And there is a difference. You can have bifidobacterium, but the different kinds of bifidobacterium will have different capacities in terms of what they can actually do in the metabolites they produce. So if you say bifidobacterium, yes, I can give you a general idea that this is what they'll do, but if you could have bifidobacterium as a predominant species and someone else might, but your bifidobacterium will do something entirely different to what theirs do. So when we talk about, say for example, your 16S testing, we can test to a genus level, but we can't test to a species level. So we're guessing when we tell you what those bacteria can and can't do. And it is a guess, it's an educated guess based on what most of the population would be, but that variation from patient to patient is going to be huge. If we take the level up from that, which is effectively what we're now starting to see come through, many, many years ago, it would have been 10 to 20 grand per test. Now we're looking at 300 to 500, depending on what level of testing you want. That's our shotgun sequencing. This is where we're actually able to tell you exactly what species are within that genus in your microbiome. That will give you a level of the alpha diversity as well, and you can get alpha diversity tested at a genus level or at a species level, and they are going to give you different information. They will look different. We can get a relative abundance of the different bacteria, and once you move to shotgun sequencing, you are also getting the functional potential, so they can say, okay, based on the genes that are in there, what capacity have you got to produce hydrogen sulfide, to produce butyrate, to produce all of these metabolites rather than guessing at them? And when I say potential, I mean potential. A lot of the times you will get these tests back and you go, oh, they've got high hydrogen sulfide. No, they don't. They have a high capacity to produce hydrogen sulfide given the right conditions. You might have an extremely intelligent child, yeah? Really high IQ, don't send them to school, don't teach them to read and write. Are they going to excel academically? No? I can have a really high capability to produce hydrogen sulfide, but if I don't feed the bacteria the right kinds of prebiotics in order for them to produce hydrogen sulfide, are they going to produce it? No. So when you are looking at your testing results, make sure that you are looking at not just the potential, but whether or not that is actually correlating with the symptoms you're getting from your clients. Where are we going from there? Not just okay, what functional potential do they have, but they are now starting to develop the means by which we can then access, are they producing those metabolites? So you'll see things like PCR and metabolomics coming through where we can actually measure the outcomes of what these bacteria are and aren't producing. Some of them we can now measure already, but getting hold of the testing can be a little bit difficult. But this is what you will start to see coming through eventually. So once we've measured the microbiome, what do we want to do with it? Everyone goes, okay, I want to fix the microbiome, I'll throw probiotics at them. Great idea? No, not necessarily. If we want to fix the microbiome, throwing probiotics at it as a temporary measure is fantastic. It will fix symptoms quickly, but it is not long term. What is influencing their microbiome is their diet, the kind of fibre that they're having, their weight, if they've got weight issues. Are they having fermented foods in their diet? Are they using antimicrobials? How many times a day is this patient touching hand sanitizer? Because that is going to influence their microbiome. How much time are they spending outside in the garden in the dirt? And then are they coming in and scrubbing their hands with hand sanitizer? So all of these things are going to have far more impact longer term on their microbiome than taking a probiotic. And I can say that as someone who's worked for the pracky companies and their raw material suppliers, yeah? The best way to influence their microbiome long term comes back to diet and lifestyle. Short-term and long-term dietary regimes, physical activity and lifestyle have been clinically proven in studies to alter the microbiome. This is not we're throwing guesses at it. There are full clinical reviews on this stuff, and I spent 2.5 years going through them. There's a lot out there. So when I look at a patient's microbiome, the first thing I look at is their microbial diversity and the richness. The second thing I go looking for is where is the correlation with the specific diseases here? Because obviously these bacteria have functional capacities, but where am I actually seeing the problems? Because that is going to tell me where I need to focus. The next thing I look for is, based on the particular patient sitting in front of me and what they are therefore going to be prone to, where is my preventative health approach with this? What do I need to focus on, not just from a functional perspective right now, but from a preventative medicine perspective? What do I need to be teaching them to get this on track? So, in terms of then influencing the microbiome, you will see here, and you guys can get a copy of the slides if you want, so you can see it in more detail, and it's in the references at the bottom if you want to access the original article. The difference a diet makes is massive. The majority of research in terms of beneficial impact on the microbiome is associated with the Mediterranean diet. Without a doubt. We get increase in things like your short-chain fatty acid production, your secondary bile acids. The increase in dietary fibre gives us improvements in pectin degradation. It's basically across the board, without a doubt, the best place to start for the majority of patients. If we look at things like a high protein or carnivorous diet, we get bacteria that are associated with an increased production of TMA and conversion to the liver TMAO, and then you get increased inflammation, heart disease, those kind of things. So what we can see is that, okay, we all know these are the health associations, but now you can see where they're actually coming from. Does that make sense? Yeah? Western diet, what most of us will probably see a lot of in clinic, is not good news for our microbiome. A lot of the bacteria we get are actually coming directly from the fruit and vegetables we're eating. So if all we're eating is processed food, there's not much of it there. We are seeing a reduction in your microbial diversity. We're seeing a Bacteroides dominant signature. Again, we're talking genus level, but overall, that's generally not a good thing. We see a reduction in the fibre and therefore also a reduction in short-chain fatty acid production. Again, that high red meat content, the increased production of TMA converted to TMO in the liver, and the increased risk of cardiovascular disease to go with it. So this is what we're not aiming for with our clients, and that's why. Another really fascinating one that I think is probably a little bit controversial for some of the old school practitioners out there, gluten restriction. A lot of us were taught fantastic for the immune system and better for inflammation. That is not necessarily the case. What the studies are now showing is that yes, where you've got someone who has gluten sensitivity and celiac disease and they swap to a gluten-free diet, there are some benefits, but there are also complications associated with that, including things like, so your benefit would be things like if you've got someone who is gluten sensitive, a reduction in microbial dysbiosis. Yeah? So there's less abnormalities and a more stable microbiome, a healthier microbiome. If, however, you've got someone who is not gluten sensitive and celiac, and we give them a gluten-free diet, we're getting an increase in the risk of heart disease. Potentially because of the less consumption of whole grain. So this is when you're seeing gluten-free diets that have effectively, they've replaced wheat with highly processed foods, and you might as well be putting them on a Western diet that is gluten-free. If you're replacing it with high fibre, whole foods, different question, those studies have not been done, or if they have, I haven't seen them yet. In a healthy cohort, putting them on a gluten-free diet, and I mean a standard gluten-free diet, not your naturopathic holistic version, for 4 weeks led to a substantially different microbiome and reduction in several key beneficial microbes. So the key summary of this is if you're going to go a gluten-free diet with your patients, please think about it first and don't just do it across the board. And make sure that you educate them on gluten-free doesn't necessarily mean no whole grains, and where are they then going to replace those fibre sources from. This was a really interesting study done in women with autoimmune thyroiditis, and they found that after 4 weeks they had increases in things like your Prevotella and your proteobacteria, and reductions in bifidobacteria was probably the big one that most people would be familiar with, and that indicated an increased risk of inflammation. This is not measuring actual inflammatory levels in the body, but this is the potential for the bacteria to produce that. So, if we want to increase diversity, we know that variation in our diet will do it. This was a study looking at diet diversity in the elderly. They did 445 Japanese people, and what they found is that diet diversity contributed to the diversity of the microbiome. Also led to increased short-chain fatty acid production, or increased potential for short-chain fatty acid production from those bacteria. Yeah? Other ways to go about improving microbial diversity is to improve the level of the variation in prebiotics that we're actually supplying. So what are we feeding our bacteria? I cheated a little bit. I didn't want to go through and do all the fiber sources and all the prebiotics for you. So having done all the research behind it or been involved therein, this is one of Microba's handouts. We can see there the kind of things that we would feed our bacteria if we want to improve IPA levels in our patients. You would be looking at ellagic acid. If you want to increase the acetate producing bacteria, you're looking at things like pectin, resistant starch, and inulin. If, for example, we want to reduce the pH, we reduce the resistant starch, or we increase the resistant starch. If you want to reduce your hexa-LPS, increase the amount of GOS in the diet. So these are all things that they're not proven in humans. The majority of them have been done in either animal or culture work, but this is how we feed the bacteria that are actually going to improve those levels for you. Again, you can use microbiome testing to know where their bacteria is at in their gut, or once you've been doing this for a while, if you don't have a patient that really needs to see the numbers on a page, a lot of this can be done through symptom management. If you've got a patient that perhaps is not responding to normal symptom management, your microbiome testing can actually add in and bring some insights that you may not have actually seen from a clinical perspective. Environmental impacts, I love this one. This is a kids' play grow study. They looked at children, giving children more time in nature. What they found were improvements in certain bacteria within their gut. They found improvements in the fecal serotonin levels, less anger, less irritability, and overall better outcomes for those children. There's another really interesting study that was actually done in adults that's spending 5 to 10 minutes a day with your hands physically in the soil getting dirty and then leaving it there for a little bit, not scrubbing it off, actually had massive improvements in their overall microbial diversity. So again, these are things that we and our patients can do that not only gives them more connectedness to their food, but also better gut microbiome. Things we wouldn't expect, they're not expensive. If anything, it will save them money because they can grow their own. Exercise and microbial diversity, this is a really interesting one because the data is a bit all over the place. What we do know is that the fitter you are, the better cardiovascular health you've got, the better your microbial diversity is going to be. However, short-term impact of exercise is a little bit all over the place. So as you're starting to actually build physical activity, the results are a bit split. Longer term impact, it is beneficial. So don't expect if you've taken a patient and told them to go do mass amounts of physical activity, that is then going to show up as beneficial in their microbiome testing immediately. Okay, weed, seed and feed. For the old school practitioners, we grew up on metagenics protocols that were, if you have a patient with gut dysbiosis, weed, seed, feed. Weed, get rid of all the bad bacteria. Seed, provide the prebiotics for the good ones to grow. Seed was provide the good bacteria, and feed was give them the chance to grow and make sure you've got the right prebiotics. That is outdated. Yeah? Badly. So when we look at whether or not we need to go through protocols like weed, seed, feed, the first thing that we need to look at is, are there pathogens or potential pathobionts in that client's bacteria? What do I mean by pathogen? Pathogen is something that is actually fully detrimental. A pathobiont is a bacterium that has the potential to cause damage given the right circumstances and given enough of it. So some of our bacteria may be flagged as potentially pathobiont, but they're only detrimental. You only want to remove them if they're actually at a level of causing problems. Do we need to remove them entirely? No, not necessarily. We just need to remove the situation that's causing them to be problematic. Remember how we talked about functional potential? They only actually produce that pathobiont effect, the negative effect, in the right environment. If we change the environment, we change their outcome. So, what we consider when we're looking at whether or not they have that pathobiont activity, we look for things like headaches, we look for nausea, reduced appetite, abdominal bloating, diarrhea, constipation, dyspepsia, flatulence. Any of those are red flags that there is some kind of pathobiont or pathogenic effect in the microbiome. Whether or not we're going to weed out those pathogens or pathobionts, look at the urgency. How severe are the symptoms and how quickly do we need to fix them? And we look at specificity. Are there any herbs or supplements that can specifically target that particular pathogen or pathobiont without adversely impacting everything else? There are certain herbs in our repertoire that are more specific or selective than others. And I'll give you some examples in a second. So if we can use diet, lifestyle to modify that pathobiont effect and to modify the environment they're growing in to change the outcome, fantastic. If it's more urgent than that and we need to change it quickly, be selective in what you're using to kill off those bacteria to make sure you're not killing off all the good stuff too. Make sense? So in terms of our antibacterials, garlic is an awesome one. It is actually quite specific, comparing its effect, for example, on E. coli versus lactobacillus adversely impacts the E. coli without as much adverse impact on your acidophilus. It also is known to actually increase your bifidobacterium in the testing as well, and we've seen that studies done on mice. Again, these are not humans because it's a lot harder to test in humans. This is all animal and in vitro, and I've put which one of those it is for you on the side. It is specific to H. pylori, E. coli, Mycobacterium, and B. fragilis. So this is one we can choose if we know we've got issues with those. You can add it in from a dietary perspective without adversely impacting our good bacteria. Nigella is another one that can have beneficial effect, very specific to things like Candida and antibiotic resistant strains of Candida without having as much of an adverse impact on other areas. So if we look at summary, first and foremost, I would say diversity is key. And that is not just diversity in terms of your microbial diversity, it is diet, it is looking at the lifestyle factors that are actually then introducing the bacteria as well. So we look at food, we look at lifestyle, and we look at the environment when it comes to the microbiome. They do not exist independently of each other. You can't treat what you don't know about. Fantastic, if you've got a client that's come in and everything presents as seen, you can work with them on that and it's having the effect you want, great. If you are not getting results, consider actually testing what's going on because it is possible there are things you don't know about. I have clients that I have worked with for 2 or 3 years who I have eventually managed to go, okay, you really need to actually look at getting this done because you're not getting the results. They get the microbiome testing done, you look at it and go, there was nothing in their clinical picture that would have told me that was what was causing the problem, but all of a sudden you can fix it very easily. Or it may be a case of you've seen something or no, they need referral for that. Then in addition to that, when we are looking at managing the pathogens and the pathobionts, definitely consider whether or not the antimicrobials or the antiparasitics are actually necessary, or can you do it with diet and lifestyle first. If there are herbals or nutritionals that you're going to use as antimicrobials or antiparasitics, please do it selectively. Choose the ones that are most specific without detrimental impact on that person's bacterium, and make sure that you're refeeding the diversity afterwards to help it recover. Questions? Yeah?
Two if I could.
Yeah.
First one is, I'm a nutritionist, so is there any particular response that you get back from a microbiome test that you go, well actually we should really be using herbs for this indication, or is there really just a slightly different perspective that us nutritionists need to take because we don't have herbs as a...
No, I think as a nutritionist, a lot of it can be done with regulating the kind of fibres and stuff you're feeding them. So if you've got pathogens and pathobionts, yes, it is a lot easier in some cases to treat with antimicrobial herbs, but as I was just saying, in some cases that's not actually the best way to go because they can do damage at the same time. So I would actually be more inclined to go the nutritional approach first and be treating with your fibres and the dietary aspects of what I could use to reduce that. And if there are a lot of the companies that actually provide the testing will provide you with nutritional recommendations of what actually can be used to increase or reduce those. I know the work we did with Cobiome was actually putting together a literature summary so that you could see the level of evidence behind each of those recommendations. So I would definitely nutrition first.
And to add to that, is there any training that you would recommend for people who want to dig into this a little bit more? Is there any particular companies who do great training in how to...
Look, a lot of the companies do great training. I would definitely say with where the research currently sits, I would be looking at companies that are using shotgun versus the 16S. I think Jason Horolak has some really interesting training. Cobiome have put together some fantastic stuff as well. But it's more about the kind of testing methods you're looking at and having a touching base with each of them, work with their trainers to see what you're actually most comfortable with in terms of how those companies communicate and what you're working with for your patients. Any others?
Questions?
What kind of tests do you actually recommend in your point of view? Because I use a lot of metabolomics, but sometimes probably you should recommend something a little bit more less specific or something that you start with, because I use biochemistry and metabolomics. So any advice that you can give?
It really would depend on what the client's presenting with. My clinical practice, I do a lot of overweight, obesity, just both because that was the area I did my PhD in and also the area where I live, and just where clinics built from over the years. So I tend to do a lot of, if I'm getting resistance to actual changes, I will look at both the shotgun sequencing with the microbiome, but I will do some of the inflammatory markers, the gut dysbiosis and stuff with it as well. I haven't done a lot of the metabolomics work because I find that for a lot of patients at this point, it is probably more on the expensive side. I think that will come down over the next few years in the same way that we've seen microbiome testing pricing come down as it becomes more readily available and the volume that they're going through is a lot higher. But yeah, for me, I tend to use your shotgun sequencing, and then if I need to, I'll add other factors into that. If they've done a lot of travel, absolutely look at your parasites and stuff as well.
For sure. No, it's because I work with more autoimmune disorders. So
yeah. So you're probably looking at path markers and stuff in addition to that as well. But I would definitely be looking at the microbiome profiles and then probably more towards your bloods and your metabolomics if that's, yeah. Over this way.
You haven't mentioned probiotics in your treatment. Do you use many of them?
I do if it's urgent. Having worked for practitioner companies and for the raw material suppliers, I'm very, very cautious of promoting different companies when I present. I think they all do enough of that themselves. Yes, I would use them if, for example, I've got a patient who I'm getting either resistance in change or their symptomology is that bad that it's going to make it very difficult for me to implement any dietary changes without fixing some of the symptoms first. Then I would use them as a temporary measure, making sure that my client is well aware that it is a temporary measure. It is to fix the symptoms so that we can then fix the underlying cause. It is never a one-stop shop and ongoing, unless obviously there's some kind of underlying disease condition that isn't necessarily fixable that is then used as a management plan. But by preference, no, I would much prefer to be working with the other stuff. I find longer term, it's more effective. Assuming you don't have a patient that's a pop a pill mentality and only likes to work with the supplements. There's that whole thing of there's certain patients that come in and they just want to walk out with the bottle because they feel better because of it. Those ones, absolutely, you could use probiotics, but definitely make sure you're explaining to them that that's only a temporary stopgap, and if they continue to eat and live the way that they currently are, it's not going to fix anything longer.